Hi, my name is Tanner Stevenson. I'm a sixth-year grad student of neuroscience at UC Davis in the labs of Tim Hanks and Rishi Chowdhury, and I'm studying the role of dopamine in behavioral flexibility. Before attending grad school, I received my bachelor's in biomedical engineering from Cal Poly San Luis Obispo, and then I worked as a software engineer at Agilent Technologies for 4 years. I got into neuroscience research because I've always been fascinated by the fact that all of our experiences, thoughts, feelings, memories, and behaviors are all manifested through the firing patterns of various networks of neurons in our brains. I'm particularly interested in understanding how the neural circuits responsible for controlling behavior can flexibly, yet stably, change their patterns of activity to drive different behaviors in response to changes in our internal state as well as our environment. These changes can occur both in the long term through learning mechanisms, as well as in the short term through what I term motivational mechanisms. Contrary to popular belief, the neuromodulator dopamine has long been linked primarily to learning and motivation, not our experience of pleasure. However, its precise role in each of these processes is still under considerable debate, including whether these are separable processes altogether or different outcomes of the same underlying computational principle. Further, there are multiple dopaminergic pathways. The nigrostriatal pathway from the substantia nigra pars compacta, or SNC, to the caudate and putamen, or the dorsal striatum. The mesolimbic pathway from the ventral tegmental area, or VTA, to the nucleus accumbens, and the mesocortical pathway from the VTA to the prefrontal cortex. These pathways have primarily been studied in isolation, and thus have been proposed to serve distinct functions. Yet, they seem to respond in similar ways, especially in response to reward and its omission. These observations have led to the theory that dopamine primarily transmits a reward prediction error to the circuits responsible for driving our behavior. Reward prediction errors essentially signal a mismatch between an expected outcome and the actual outcome, and are the computational backbone of reinforcement learning and AI. For example, if a reward is unpredicted, say after this tone, there is a positive RPE. Then, if a reward is predicted at the time of the tone, you have a positive RPE, and then no RPE at the time of the reward. And if a reward is expected, but is not delivered, you get a negative RPE at the time of the omission. These prediction errors can then be used to update behavior in the short and long term. Though much of the data collected to date aligns with this theory, there's recently been a surge of data that seems to contradict it, thanks to new experimental tools to measure and manipulate dopamine release in rodents. I am using one such tool, a fluorescent dopamine sensor, to record dopamine release simultaneously across all these dopamine pathways as rats perform 2 different tasks requiring flexible behavior. With these data, I will then characterize the precise ways that dopamine signaling is either consistent or distinct between regions, as animals are freely moving and adapting their behavior to the context of each task. The goal of my work is to provide constraints to existing theories of dopamine function, and hopefully to inspire new theories about what this crucial signaling molecule is actually doing. Thank you.